Metformin is a drug that has historically been used as a way to manage type 2 diabetes as it increases the body's response to insulin and decreases the amount of glucose in the body. However, recent studies have explored the potential for metformin to be used as an anti aging intervention as it can affect various processes throughout the body.
There are many processes in the human body that leads to aging, and metformin can affect many of these mechanisms. Today we'll go over just a couple of them. First, as we get older, advanced glycogen end products, also called AGEs, build up in our bodies. AGEs are formed when glucose interacts with collagen, and they're associated with wrinkles, hearing loss, vision loss, and decreased blood vessel elasticity. The more glucose in the body, the more AGEs are formed. Metformin, however, reduces the amount of glucose present in the body, therefore reduces the amount of AGEs, potentially leading to increased health span. Oxidative damage also increases with age. Oxidative damage occurs when molecules in our cells lose electrons and turn into unstable free radicals, which can cause damage to our cells and DNA. Our body has natural defenses against oxidative damage, such as antioxidants. However, as we age, the amount of free radicals increases and the body's ability to resolve these issues decreases. Metformin slows the creation of free radicals and thus decreases oxidative damage. This allows the body's natural defenses against oxidative damage to work, decreasing the rate of aging and potentially increasing lifespan. Finally, our bodies have energy molecules called AMP and ATP that help manage our cell functions. AMP and ATP convert back and forth, so as one of these concentrations decreases, the other one increases. When there's plentiful ATP, this leads to cell growth through an activation of the TOR pathway and the inability of the body to get rid of damaged cells, also known as autophagy. Growth is beneficial in early life but causes aging later in life. Metformin increases AMP levels, causing the signaling protein AMPK to inhibit the growth pathway of TOR, as the body wants to conserve energy. This also results in an upregulation of stress response genes and also upregulation of autophagy. This can increase lifespan as our body can handle stress and damage better. Through these mechanisms, metformin may be able to increase health span and lifespan, and scientists have put this theory to the test.
This first study investigated how metformin affected the health span and lifespan of C. elegans worms. In one of the experiments, researchers had a control and mutant DAF16 worm group. DAF16 is a gene that's typically necessary for the TOR pathway, so if added, TOR was inhibiting these mutants. Then half the worms in both groups were subjected to metformin and the other half were not. These experimental results showed that metformin was able to extend the life and health span of both groups of worms. This suggests that metformin wasn't acting only to increase the life and health span through the TOR pathway, it was probably through other mechanisms as well. However, additional research is still needed to see if these effects can translate to humans. It also raises the question if metformin should be administered even if we don't know these other pathways well enough.
This study investigated the effects of metformin on mice, examining two different strains under four conditions: a normal diet, a calorie restricted diet, a normal diet with a low dose of metformin, 0.1%, and a normal diet with a high dose of metformin, 1%. The results showed that mice on the low dose of metformin experienced both extended lifespan and improved health, such as less lens opacity, a sign of delayed onset of cataracts. However, the mice on the high dose of metformin had a roughly 14% decrease in lifespan, highlighting the importance of dosage, as too much of metformin could potentially be harmful. The study also found that gene expression patterns in mice treated with metformin closely resembles those on a calorie restricted diet, which is a known anti-aging intervention. These findings suggest that metformin has potential as a treatment to delay aging and age-related diseases, but also that dosage is a critical aspect of its use.
This final study looked at the relationship between long term metformin use and cognitive function in older adults with type 2 diabetes. The study had two main groups: metformin users and non users, with the metformin users also divided by treatment duration. The global cognitive function of these participants was examined and followed up again after 4 years. This finding supports the idea that long term metformin use could delay cognitive decline in Alzheimer's disease. However, the data relied on participants self reporting their metformin use, and it did not account for dosage levels, which, as discussed previously, may be an important factor. Also, these results can't be generalized to all people, as it was only tested in older people with type 2 diabetes.
In model organisms, there's evidence that metformin can increase the health span and lifespan. However, the question remains if these benefits will translate to the general human population. More research is needed to validate metformin's positive results and determine the logistics behind how metformin's dosage would work within humans. Luckily for us, there's a study called TAME, targeting aging with metformin, that's currently looking at how metformin affects the aging process in 3,000 people. If the results of the TAME study show that metformin increases health and/or lifespan without unwanted side effects, we think that this could potentially be a great anti-aging intervention. However, until then, we can recommend with certainty that metformin should be used to combat aging.